Hello, in this video we will discuss about the lipoprotein metabolism. So what is lipoprotein? Lipoprotein means lipids contain protein. Lipid is the fat and containing cholesterol, which can be dietary cholesterol or synthesis cholesterol from the liver. So the micelle is the precursor before the digestion, which contain maximum fat but minimum protein, while the chylomicron after the digestion will become a more protein as compared to micelle, and VLDL, very low density lipoprotein, contain a fat more as compared to protein, and IDL basically contain more fat but as compared to VLDL is less. So in this way, the LDL is low density lipoprotein, which contain a more fat, but the percentage of the protein is more. For example, the 40%, while the HDL and HDL empty contain half protein and half lipids. So let's begin to understand the fats and dietary fats and cholesterol. So what is the significance of the cholesterol and the lipoprotein? That is we will discuss also. And the lipoprotein is basically our need for the synthesis of hormone, and cholesterol also play important role for the steroid synthesis and hormone synthesis for the male and female identification. So this is the fats, dietary fats and cholesterol. When enter through esophagus, will go into the stomach. After go and into the duodenum, the digestion will begin through a lipase enzyme will produce from the pancreas, and the gallbladder produce the bile pigment and bile salt for the emulsification of the lipoprotein, emulsification of the fat. Triglyceride will convert into the micelle. So let's begin to understand. This is the lumen and this is the bigger picture of the duodenum. This is the enterocyte brush border cell, and it's contain enterocyte, and this is the GLUT4 channel which that will be allowed to sugar move inside with a 2 sodium ion, that is known as symport transport, and glucose will move inside into the blood circulatory system after the absorption. So the insulin will trigger by this mechanism through hyperglycemia. So these lipoprotein droplets, which lipid droplet, will convert into the micelle due to the gallbladder which produce the bile salt. So the cholesterol will also absorb, and apoprotein B100 will be synthesized through transcription to translate into the enterocyte and addition. So these monoacylglycerol and fatty acid combine together to form a chylomicron. This is the initial entry through lacteal duct to the blood circulatory system. So the lipoprotein lipase enzyme, the hydrolysis of triacylglycerol in vessel. After this, the free fatty acid and glycerol will enter into the fatty acid and glycerol into the adipose tissue as well as into the muscle, and into the muscle when go, the beta oxidation will lead to produce the ATP. Beta oxidation means the breakdown of fat. So this is the adipocytes. When glycerol and free fatty acid move into the adipocyte, so this is the resynthesis of the glycerol and fatty acid combined to human sensitive lipase enzyme, and there are a lot of enzyme condensation reaction to produce this triacylglycerol into the adipose tissue as a storage. So the adipose tissue important for the store of fat in the form of triacylglycerol. After this, the remnant chylomicron after the deposition of fat will be go into the liver, and when go into the liver contain receptor, LDL receptor or scavenger receptor. This is the remnant chylomicron will go into the liver, and the extra cholesterol will be excreted from bile into the bile pigment and bile salt. While here is the endocytosis through scavenger receptor will move inside, and endosome bind together to splitting of the monoacylglycerol, and there are a lot of things. So remember, this is the emulsification through bile acid. So here is the glucose when increased into the blood circulatory system will lead to enter through GLUT transporter channel into the glycolysis to produce pyruvate, and pyruvate will enter into the mitochondria and produce acetyl-CoA. This acetyl-CoA will convert through HMG-CoA to produce cholesterol, while the malonyl-CoA also produce. This is the chart for understanding, not we are going to whole detail. So this is the statin. This statin is the HMG-CoA reductase enzyme will inhibit the acetyl-CoA conversion into the cholesterol. So it is the cholesterol inhibiting agent, that is drug. And on the other hand, this sugar when glucose will be enter also convert into the glycerol, and malonyl CoA will convert into the fatty acid. And in this way, the glycerol will combine together with fatty acid to form a monoacyl glycerol and triacyl glycerol. And this triacyl glycerol here is you can see endosome formation, and cholesterol will combine together with triacyl glycerol, enter into the Golgi bodies. After enter into the Golgi bodies, that will be modification will become a lipoprotein packaging by the apoprotein addition. This apoprotein will come from the transcription to translation from the hepatocyte. So on the other hand, this is the lipoprotein. This is the LDL or HDL. So this is HDL, high density lipoprotein, which contain a triacylglycerol inside and phospholipid layer is the outer, and it is the hydrophobic, and it contains basically apoprotein and triacylglycerol and cholesterol. Cholesterol is basically in the form of cholesterol ester formation, while on the other hand the phospholipids, and this is the droplet which that will be transport exocytosed out from the liver hepatocyte toward the blood circulatory system. And when entry, this is the empty high density lipoprotein which that will be able to pick up excess cholesterol from the tissue. So after this, when excessive cholesterol are present in the cell, which that will be pick up cholesterol, and high density lipoprotein which contain empty will be filled with the high density lipoprotein contain cholesterol and fats. And this basically high density lipoprotein will target again into the liver for again taking off the cholesterol into the liver for excretion the extra cholesterol into the bile acid. So in this way, here is the apoprotein C is used for the high density lipoprotein receptor. When endocytose will again the cycle will be occur. So this is the basically toward and away from the cholesterol for the major mechanism and metabolism. So here is you can see this is the low density lipoprotein will be recycled, move again very low density lipoprotein. Remnant chylomicron will convert into the very low density lipoprotein. And lipoprotein lipase enzyme which that will be used to produce IDL, and in this way the remnant very low density lipoprotein and adipose deposition into the adipose tissue, and LDL will be again remnant very low density lipoprotein which that will be endocytosed in the muscle cells. So this is the muscle cell contain a receptor, this LDL receptor or scavenger receptor, and this vesicle is formed, and this endocytosed vesicle. After this endocytosed vesicle, the endosome will be attached within it, and in this way the very low density lipoprotein will be split. But splitting, before splitting, the lysosomal droplet or lysosome will be combined together, and lysosome is the very important for the hydrolysis and breakdown of these lipoprotein. And after this breakdown, in this way it can be recycled, and after the endosome breakdown, and in this way the endosome breakdown the enzymatically break up of lipoprotein low density lipoprotein, and in this way the amino acid and cholesterol and fatty acid which that will be split and increase in the tissue, increase into the muscle, and cholesterol ester basically form. And remember, these cholesterol which that will be used into the plasma membrane for the increase of the fluidity and stretching of the plasma membrane for growth and development of the muscle. So on the other hand, the cholesterol is very important for the reproductive system to produce the estrogen and progesterone, as well as on the other hand the testosterone. So this is also amino acid, cholesterol and fatty acids splitting to produce ATP. So this is the whole mechanism of the lipoprotein metabolism. So I hope you make sense about these things. Thanks for watching.